Welcoming Dr. Lanxin Ji. Lanxin is an accomplished postdoc at NYU Langone, and I'm not saying that because she's my friend. You may remember her name from last year, as she won the Best Paper Award from Human Brain Mapping for work of fetal behavior in MRI. Today she's going to talk about her most recent work on fetal and infant brain iron.
Okay. Thank you, Aris, for the introduction. My name is Lanxin, and it's a great pleasure for me to present my work here. Today I'm going to talk about the brain iron from the very early stage, from fetal to infant life, and its impact on neural activity. So we have already know that iron is crucial for many developmental processes like the oxygen transportation, myelination, and is also related to many developmental disorders like ADHD and autism, so it's important to measure it. Dr. Moeller has already gave a comprehensive review of how we measure iron in the brain, and in this study we used the multi-echo fMRI data sequence to measure brain to record the neural activity and brain iron estimation at the same time. And our group has been spent 12 years in using imaging fetal brain in utero before birth for a long time. So we really curious on whether we can measure brain before birth and how it changed from the utero life. So here's how the fetal fMRI data looks like. So we can see the brain, actually it should be a movie. So actually we can see the fetal brain, the fetal is moving all the time during our scan. And so the fetal fMRI images has its unique challenges. So for example, the low signal to noise ratio, and the baby is moving all the time. There's always high levels of motion, and there's a non-rigid high intensity mother tissue interactions. So we have been spent years to overcome these challenges, and in this study, we used a deep learning based approach to extract brain from the mother tissue first, and once we get the brain out, we can estimate the T2 star and R2 star at the voxel level. And because the brain can be at multiple orientations, so we used a manual reorientation algorithm here and used the fMRI data instead of the T2 star map itself to run the normalization to transform it from the individual space to the standard space. And it's notable here that we used a stepwise normalization algorithm here because the fetal brain always changes so rapid and so much variance between ages. So we use the fMRI data to estimate the transformation matrix between ages and then concatenate them to one single matrix and apply it back to the T2 star map, and it goes to the standard space. And then we use a cutoff to exclude the extreme values in the R2 star, and it's ready for the regional and tissue-wise analysis. So the quality assurance here are quite important because we do see there are a lot of extreme values in the R2 star estimation. We will look into the data. We drop pins in the brain. We say when the signal is decay continuously, we got a good fit. But when the signal goes going up and down or down up, it leads to a bad fit, and the R2 star goes to extremely high or negative values. So in this study, we exclude those values to ensure that all the R2 star estimation in our further analysis are quite reliable. So here's the results. The final sample includes a longitudinal data set, 86 fetal scans from 52 fetuses. And we do see significant increase of iron cross-age across all tissue segments, including white matter, green matter, left hemisphere, and right hemisphere. And in our regional level analysis, we see 21 regions exhibiting a significant increase in R2 star with age during this fetal period. And it's interesting that this region spanning many different brain lobes from the cerebellum to the occipital, parietal, temporal, and subcortical regions. We see there are two possible notable effects here. One is that we don't see any significant result in the frontal area. So it is quite reasonable because the frontal region is supposed to be develop later. And the other effect we see here is the motor cortex and cerebellum demonstrated the most pronounced age-related increase in brain iron. So we infer that it may reflect that more rapid development in these regions during this early period. So the next question arised here is whether there's a sex difference in brain iron development before birth. And the answer is yes. We do see when we add the age by sex term in our regression model, we do see there's a significant effect of sex in the left cortical gray matter and the left white matter. And in the right side, we also see the same trend. And in the regional level, the only region that highlight here is the left insula. So we know left insula is a key region in emotion regulation. So why it's important, why it's different in sex is an interesting question to think. And the next question we're curious about is, now we know how it changed before birth, so how it changed across the birth. And fortunately, we do have this data. In this data set, we have to follow up the fetuses until their birth. So in later explorer analysis, we include 86 fetal scans and 62 infant scans. We plot the trajectory across the lobes. First, in the parietal lobe, we do see rapid change both before and after birth. There is a constant increase of iron during this perinatal period. And interestingly, in the occipital and temporal lobe, they shared a similar pattern that there's accelerated increase around the birth event. And it goes pretty fast before birth and slowing down after birth. And interesting, again, in the frontal lobe, we see a quite flat trend, which may mean that the frontal is not the region at this period. So it mirrored what we see in the fetal earlier analysis that we presented within the fetal data set. So the next study I'm going to talk about is how iron development relates to the neural activity. So we have already know that iron is coupled with the cognition. And there's a bunch of studies showing that resting state fMRI is related to the cognition. So it's naturally to think how iron and resting state fMRI related to each other. And we believe that there is an underlying biological mechanism is that the iron is critical in many metabolic and energetic processes, and this process are also related to the neural activity. So in this study, with a better quality, image quality in the infant data set, we run a voxel level conjunction analysis between the iron map and the both signal. So as I mentioned, we estimate the iron with a multi-echo fMRI data. So the fMRI data, the both signal and the iron estimation are acquired simultaneously. So in this study, we used the linked independent component analysis, which may be familiar to you. It's a conjunction analysis approach that is designed to reveal the hidden relationship between images from different modalities. So for iron proxy, we use the R2 star map, and for the neural activity, we use the ELF, which is amplitude low frequency fluctuation. It is a summation of the neural activity within a specific period of specific frequency band. And as shown here, we have 5 components stands up. So we can the blue is showing the iron deposition. The right is the ELF. Is from the ELF, and so we can see they're quite pretty much co-localized to each other across all components. And the first components pretty stand out because it is much more extent than others. And against the almost the whole brain iron deposition, we see the function distributed comprise the primary regions of default mode network. So we know that default mode networks is always in a high metabolic demand, and it should be related to iron. So we kind of infer that the whole brain iron increase may support the early development of default mode network. And we further explore whether there is a developmental effect between across the components, and we do see the only significance falls into the first component, which means the iron in these regions are iron and both iron and alpha increases in these regions are increasing as age advances. And yeah, interesting thing to note here is that if we see the age effect with the unimodal analysis within either of them, we cannot see this picture. So brief conclusion of this study. So we plot iron across births, and both before and after births, and we do see there's a significant increase across the whole brain rather than the subcortical regions that we always believe which is important. And we do see there is a sex difference that females accumulate iron faster than males in most of the, in almost every tissue segments and the left insula. And in infants, we do see that the brain iron is directly related to the neural activity, and they're quite co-localized. So we do say, so in general, this work we think is a basis of a general developmental framework. So we can imagine that from this work, we can do a bunch of interesting things. So here I provide a previous report conducted by Campbell and colleagues. They found the maternal mental health can affect the cord blood firing in their offsprings. And there's a sex difference. And within our group, Dr. Manuel is also exploring the effect of maternal inflammation on our brain iron data. So she does say there's a difference. So we can imagine, like, there are a bunch of opportunities to look at the environmental and context effect of brain iron development during this period. Yeah, so that's it. Thank you. It is a teamwork for sure, and thank you, all the team members and the PI of my lab, like Dr. Marian Thompson, and welcome to stop by our posters. So I have another poster presented this afternoon, which is on a different topic about edge-centric functional connectivity in fetal brains. Yeah, feel free to stop by.